Yeah, hello everyone. Myself Dr. Kanagaraj, Head in Biotechnology, Rathinam College of Arts and Science, Coimbatore. So today I am going to present about the polymerase chain reactions. So polymerase chain reactions is a help of the enzymes to amplify the DNA fragments. Our interested DNA fragments we are going to amplify based on the reaction of polymerase. So this reaction first invented by Carey Mullis in 1983. So based on the invention, he has received the Nobel Prize in 1993. This reaction components, we need a DNA sample is a template and primers, DNTPs, then Taq polymerase, then buffer. So Taq polymerase we are isolated from the Thermus aquaticus is a microorganism, the thermostable microorganisms is isolated. So this mixer we are keeping in the PCR machine, we call the thermocycler. So thermocycler means it's maintaining the temperature. The reaction carried out the following process. So initial the denaturation, annealing and renaturation process for the reaction process. So this denaturation, initial denaturation, it will take 4 to 5 minutes for the 94 degrees Celsius. So at that time, so all the DNA molecules, double stranded, it will convert into single stranded. Then after that, it will bind to the annealing, is a primer binding process. So the annealing process is depend on the primer for the temperature, so 55 to 65 degrees Celsius annealing. So during this time, the primer it will bind to the DNA fragments for the interstellar size of the fragments. Then it will extension for the 72 degrees Celsius for 1 minute. So it will the cyclic process will carry out for the 30 to 35 cycles. Okay, this is the process for the PCR reactions. Okay, so this PCR reaction, after completing the PCR reaction, we are going to run the agarose gel electrophoresis. Will find the quality of the amplified product. So this PCR reactions. This PCR reaction we are going to apply the various clinical aspects. The applications for the PCR process, the diagnosis of the disease. So what are the disease we occur, so based on that we can able to identify the particular disease and genetic disease, selective DNA fragment isolation, and apply for the DNA fingerprinting technology, then checking of the contamination of the microorganism in the food product, then sex determinations, a male or female sex determination, then quantification of the nucleic acid. So how much amount of the nucleic acid presence in our genome, the estimation of the gene expression. So how much amount of our gene it will be excess for our product, then validation of the cell lines for the animal cells. So different kind of animal cell available. So based on the PCR technique used to validate the quality of the animal cells, then cloning for the PCR product. So our interested fragments amplified and cloned to the genome. The plant research and agriculture process, so hybrid variety of plant research we are going to apply for this technique. Then animal cell culture research, then PCR as a antiviral therapies. So nowadays they are very familiar with the COVID-19 identifications of the viral process. Then development of the markers. So different kind of markers, DNA fragment markers available, we can able to develop. These are the application for the PCR techniques. Thank you.